Methods. Ethics statement. The study was focused on vertebrates, birds of paradise, but used museum specimens, physical and media, so no IACUC protocol was required. Behavioral complexity. We quantified the behavioral complexity of courtship display behaviors for the birds of paradise by scoring field-recorded video clips of 32, 80% paradisiae species, primarily from the Macaulay Library at the Cornell Lab of Ornithology. In total, we watched 961 clips from 122 individuals, totaling 47,707.2 seconds, approximately 795.12 minutes, mean clip duration of 49.64 seconds. Courtship display behavior is highly variable among bird of paradise species, necessitating broad behavioral categories to facilitate investigations of behavioral evolution. Specifically, One of Us, CDD, blindly evaluated video clips of male birds of paradise displaying species-typical courtship behaviors using a customized ethogram of behavioral units that enabled us to quantify all state and event behaviors exhibited by all species of Paradisidae. S4 table. Data collection. To record courtship display behaviors, we used a customized version of an open-source behavior logging program. Additionally, we created a customized keyboard that allowed us to quickly and accurately record the start-stop times of all duration behaviors, as well as the instances of all event behaviors. The combinations of different behavioral categories throughout each clip allowed us to generate sequence data of distinct behavioral elements. Measures of sexual display behavior complexity. Courtship displays can be broken down into distinct behavioral elements and the transitions between these elements. We investigated the number of unique behavioral elements, behavioral richness, in a given time period, as well as the Shannon entropy of these behaviors, behavioral diversity. Shannon entropy provides a measure of information encoded in the behavioral displays, and we converted Shannon entropy scores to their numbers equivalents. Shannon indices were chosen specifically because they are the only measures that give meaningful results when community weights are unequal. As previously described, the numbers equivalent for Shannon entropy values has the readily interpretable property whereby a value of 2x would indicate a behavioral sequence with twice as many equally well-represented behaviors as a sequence with a value of x. In the context of behavioral displays, birds that use many unique behaviors and spend roughly equal amounts of time performing each display element, increased evenness as a proportion of time, will have higher diversity scores. Sliding window analysis. The number of courtship recordings available was highly variable across species of birds of paradise, S3 table. To reduce the influence of sampling intensity on our overall behavioral analyses, we used a sliding window analysis to evaluate similar time windows for courtship display complexity across species. Specifically, we used a sliding 50-second window, chosen as the minimum duration, resulting in relatively stable individual behavioral complexity scores across all clips for a given individual to identify the specific 50-second period of maximal display complexity for that individual, and incorporated the resultant complexity scores for this interval in our analysis. Individual scores were then averaged to obtain species-level estimates of signal complexity. Collectively, our approach minimizes the influence that variation in recording time and clip duration has on species-level behavioral comparisons. Our results and interpretations are robust to the choice of different window sizes between 10 and 60 seconds. Color complexity. Image collection. We collected images from 393 Birds of Paradise Museum specimens, S7 table, housed at the American Museum of Natural History. Specifically, we took raw format images of adult males from 40 Bird of Paradise species under standardized conditions using a Canon 7D camera, with full spectrum quartz conversion and fitted with a Novaflex R 35mm lens. Illumination was provided from two eye-color arc lamps, diffused through polytetrafluoroethylene sheets 0.5 millimeters thick. These arc lamps are designed to simulate International Commission on Illumination (CIE) recommended daylight D65 illumination, though they come standard with a UV blocking coating, which we removed prior to use. Additionally, for every specimen position, see below, we used filters to take 2 photos, one capturing only UV light 300 to 400 nanometers and one capturing wavelengths between 400 and 700 nanometers. To simulate a variety of viewing angles and increase the likelihood of capturing relevant coloration from bird specimens, we took photographs of each specimen from 3 viewing angles: dorsal, ventral, and ventral angled. Specifically, each specimen was photographed from above while it was flat on its belly, dorsal view, flat on its back, ventral view, and angled 45 degrees on its back, rotating the frontal plane along the vertical axis while keeping the head oriented in the same direction as the previous 2 photographs. The angled photograph was taken to increase the likelihood of capturing some of the variation made possible by iridescent plumage. Image processing. UV and visible spectrum images were used to create standardized, i.e. channels were equalized and linearized, multispectral image files for each specimen position using the image calibration and analysis toolbox in Image J. Avian color vision. After estimating the color sensitivity of our camera lens combination, we generated custom mapping functions to convert image colors to stimulation values corresponding to an avian visual space. Birds of paradise are inferred to have a violet-sensitive visual system, and the curl-crested Manicode, Manicoda comri, and magnificent riflebird, Taurus magnificus, have the same amino acid sequence in spectral tuning positions 84 to 94 as the jackdaw, Corvus monodula, which is inferred to have a peak sensitivity of its violet sensitivity type SWS1 cone at 408 nanometers. This sensitivity is similar to that of another species with a VS visual system. VS is violet sensitive. The pigeon Columbia livia, SWS1 peak sensitivity equals 404 nanometers. Consequently, we converted our full spectrum photographs into the perceptual space of pigeons using physiological data, spectral sensitivity curve functions implemented in the R package PAVO, and multispectral imaging software in Image J. Additionally, we evaluated color using a visual model from a UV-sensitive passerine species, the blue tit, and found our results qualitatively unchanged. Prior to subsequent clustering, see below, we performed a median pixel blur to eliminate aberrant pixel values owing to dust on the sensor, temporary dead pixels, etc. Color clustering. Following conversion to avian color vision and noise filtering, we used a novel custom-written agglomerative hierarchical clustering algorithm to reduce each multispectral image down to a perceptually relevant number of color clusters. This clustering algorithm was developed from a more basic algorithm used previously that did not integrate luminance or thresholds when combining clusters. At the first step of the clustering process, each pixel is its own cluster. Each cluster is then compared to its neighboring clusters in the XY plane of the image within a given radius, 1 pixel initially, and composite distances are calculated based on an equal weighting of chromatic and achromatic, just noticeable distances, JNDs, using the log model. Specifically, chromatic and luminance JND values are divided by the chromatic JND threshold and luminance JND threshold, respectively, so that they are weighed equally based on the chosen threshold, and then the Euclidean distance of these two scaled values is calculated. Following distance calculations, each cluster is combined with its nearest, in composite JND distance, neighboring cluster if the composite difference is below the threshold for both luminance and chromatic JNDs. Nodes can have multiple clustering events at each pass, e.g. if cluster A is closest to B, but B is closest to C, and all distances are below the threshold, then all three will be clustered, meaning whole strings or neighboring regions can be clustered. In practice, each cluster tends to be combined with 2 or 3 clusters on each pass. On each pass, the updated mean cone catch values for each cluster are calculated, ready for the next pass. Additionally, the XY distance search radius increases with each pass, so as clusters get larger, they can also combine with neighboring clusters further away, which has the desirable effect of also keeping the processing for each pass relatively constant, i.e., there are fewer clusters on each pass, but each one must be compared to a larger number of neighbors. Clustering therefore takes place across n plus 2 dimensions, n colors plus x and y space. The code used for this clustering is provided as supplementary material, S1 code, and we have included a sample image illustrating the output of the clustering process, S5 figure. Measures of color complexity. Following color clustering, we quantified plumage color complexity using analogous indices to those we employed in our behavioral analysis, namely, we quantified color richness, the number of distinct clusters, and color diversity, the numbers equivalent of Shannon index for each view, dorsal, ventral, angled, and averaged these values to obtain individual specimen-level metrics of color complexity. In terms of color, specimens with higher richness scores have more unique colors, and species with high diversity scores have more evenly distributed colors. Influence of specimen age on color complexity measures. Aging can influence the coloration and appearance of some kinds of avian plumage, although such effects are often relatively small. To evaluate the possibility that specimen age might influence our estimates of species-level plumage elaboration, we conducted a linear mixed effect model with 2 measures of color complexity, color richness, color diversity, as the dependent variable, collection year as the independent variable, and species as a random effect. Analyzing these models revealed no significant influence of collection year on either color richness or diversity. Acoustic complexity. As with display behaviors, we quantified the acoustic complexity of courtship sounds produced by analyzing field-recorded audio-video clips of 32, 80% bird of paradise species. In total, we analyzed sound from 176 clips from 59 individuals, totaling 43,670.9 seconds, approximately 411 minutes, mean clip duration of 140.18 seconds, S8 table. The birds can generate sounds, both vocally and mechanically, in numerous contexts. We focused our analysis of recordings from known display sites or those matching written descriptions of courtship sound production. Data collection. From each video clip used to quantify display behavior, we identified a focal individual and all of the sounds it produced. Spectrograms of the audio were viewed with a frequency resolution of 43.7 Hz and time resolution of 2.31 milliseconds, and all sounds were marked in the sound analysis software Raven Pro version 1.5. Individual sounds were defined as temporally separated sound elements. Using the robust measurements in Raven, we measured the duration, maximum and minimum frequency, bandwidth, peak frequency, and peak frequency contour of each call. We measured the disorder, lack of organized or tonal structure, in a call with aggregate entropy and average entropy measures in Raven. Following detailed analysis of the acoustic parameters for all notes, we used a 2-step semi-automated classification analysis to assign note identity. In the first step, we conducted a principal component analysis, PCA, using 15 summary acoustic variables, S9 table, followed by agglomerative hierarchical clustering to assign partial note identity, i.e. note classification, based on location in PCA-based sound space. In the second step, each note was given a categorical identifier depending on the combination of 4 qualitative variables manually scored as yes/no: frequency modulation, non-harmonic structure, impulsive, stochastic. Full note identity was achieved by merging the clustered note identity with the combined qualitative categorization. Measures of acoustic complexity. After assigning identities to all notes in our dataset, we measured acoustic richness, number of distinct note types, and acoustic diversity, Shannon index of nodes within a given time period. See sliding window analysis below. As with behavior, we used the numbers equivalent of Shannon index values to facilitate more direct comparisons among samples and species. Sliding window analysis. The duration and number of available courtship-specific acoustic recordings was highly variable across birds of paradise, S8 table. To reduce the influence of this variation on species-level acoustic comparisons, we used a sliding window analysis, similar to our behavioral analyses, to evaluate and compare similar time windows for acoustic display complexity across species. To identify the time period of maximal acoustic complexity for an individual in our analysis, we used a sliding 10-second window, chosen as the minimum duration resulting in relatively stable individual complexity scores, S6 figure, across all clips for a given individual. Individual scores were then averaged to obtain species-level estimates of signal complexity. Relative complexity measures are robust to the choice of different window sizes between 5 and 50 seconds, S7 figure and S8 figure. Phylogenetic analyses. Tree. We regenerated a phylogenetic hypothesis from a recent molecular phylogeny for Paradisidae using the function phyllo.tracer in the R package PhySketch. This ultrametric, time-scaled tree was used for all downstream comparative analyses following one modification of tree typology. Specifically, we placed Lufarina superba as the outgroup to Tolaris to accommodate a revised taxonomic hypothesis. Phylogenetic generalized least squares. For each of the 6 components of courtship phenotype, color richness and diversity, behavior richness and diversity, acoustic richness and diversity, we conducted a single MPGLS regression evaluating the influence of the other elements of courtship phenotype and 2 signal environment variables predicted to influence relative investment in separate axes of overall courtship phenotype. Specifically, we included a categorical metric of display height in all models, in which each species was scored as displaying on the forest floor, in the understory, or in the forest canopy. Additionally, we included the categorical metric of display proximity in all models, where each species was scored as displaying solitarily in exploded leks or in true leks. In each model, see S1 table and S2 table, we included only like phenotype measures, e.g., including behavioral and acoustic richness, but not behavioral or acoustic diversity when investigating the drivers of color richness. All courtship phenotype measures were log transformed prior to analyses, and analyses were performed in the R computing environment using the GLS function and the NLME package, assuming an Ornstein-Ullbach OU covariance structure using the Comartins function from the APE package. Imputation. We used the Rylofars package in R to impute character values for taxa with missing data, e.g., species lacking behavioral acoustic information, S1 data. This methodology has previously been found to perform well in predicting ancestral and missing species values. In our case, we evaluated the performance of several methods to estimate missing values assuming a Brownian motion model of trait evolution, an OU model, an early burst model of trait evolution, a Pagel's lambda model of trait evolution, and a multivariate OU model. We compared model performance by evaluating AIC scores and determined that the OU model performed best. Consequently, character trait values imputed using this model were used in all subsequent analysis. Though data imputation can increase statistical power, the instances in which it might induce spurious findings are few, especially given the relatively small proportion of our total dataset, 20%, for which we imputed values. In fact, bias tends to be lower when missing data are inputted rather than omitted. Regardless, to alleviate concerns that imputed values may drive subsequent findings, we also conducted our phylogenetic least squares (PGLS) analyses on the limited subset of species n equals 13, for which we have complete data. In all cases, the findings were qualitatively identical to those reported in the main text, S10 table and S11 table.